presenter is Julia Taylor, and her research on identification of feline oral flora by biochemical and molecular investigations. So we'll turn it over to Julia.
Hi. So I'd like to start by talking about the microbiome a little bit and what that actually is. The microbiome is the collection of bacteria, viruses, and single-cell eukaryotes within the healthy individual. That individual might be a human, a canine, or a feline, but the individual nonetheless. Just to give you a few facts to kind of put things into perspective, in the human microbiome, microbial cells outnumber human cells in a ratio of 10 to 1. Astonishingly, microbial genes outweigh human genes in a ratio of 200 to 1, and a lot of scientists believe that that's actually a conservative estimate. And even more astonishingly, actually 5 pounds of the adult body weight is made up of these microbes within our bodies. So in considering the fact that we have all these microbes living within our bodies, they're not just living there. They actually contribute to a lot of physiological mechanisms, such as metabolic functions in the gut, immunological activity, immunological protections in the epidermal areas, nutrient production, even vitamin synthesis. So clearly it's very important to categorize these species and know what's there to be able to define what a healthy individual is, and then to be able to identify other susceptibilities to disease or potential irregularities with that microbiome. So the fact that we do know a lot about the microbiome and how important it is, it's kind of interesting that we actually, over 95% of it, we don't know anything about. We haven't been able to isolate or study any of these, and the reason because of that is microbiology hinges on the fact of what you can culture. And so 95% of these bacteria, they're not cultivable, or we haven't developed the methods yet. Bacteria are very picky. They really care about what media they grow on, what environment they're in, what nutrients they're exposed to, oxygen availability. The list is pretty much endless. And if you think about the complex physiological conditions that these type of commensal bacteria are living within, things get a lot more complicated. And it's harder to do that in the lab. So in this particular study, we're talking about a feline oral microbiome. And in talking about the oral microbiome, you really can't not talk about biofilms. Biofilms are that kind of fuzzy film you feel in your teeth after you forget to brush for a day. But basically, they have a sequential development. They start by planktonic bacteria adhering to a pellicle. And that provides a surface where further growth can occur, and then it gets more and more complex. And I'll talk a little bit later about the structure of these biofilms, but these initial colonizers here are vital because without them, you won't have the complex interactions that occur on top of that. And specifically in this study, you'll see that Neisseria and Veillonella are very important. So a little bit about the project background. In the 2007-2008 academic year, Dan Yeager and Vicky, I'm not going to pronounce her last name, but Dan and Vicky, they isolated they did supergingival scrapings in the supergingival area indicated by the blue arrows here of a domestic feline cat, about 12 years old, healthy male cat. They cultured this and they grew it up, and through various techniques, different culture methods, different aerobic conditions, anaerobic conditions, they were able to isolate 10 different strains, from which they selected 2, a gram-positive and a gram-negative. They named their gram-negative FO7DBA, and that's the strain that I'll be working with, or that I have been working with. They found that it was a gram-negative diplococci, facultative anaerobic, but it also grew pretty robustly in aerobic conditions. And they tentatively classified it as a Neisseria species based on a very limited amount of biochemical tests. I'm just going to give you a brief background about oral Neisseria so you can see where I came into the project and what my ideas about it were. Oral Neisseria are non-pathogenic. There's 2 pathogenic Neisseria strains, Neisseria meningitidis and Neisseria gonorrhoeae, but thankfully those aren't in our mouth, so we don't need to talk about those. They're diplococci arrangement, non-motile, and then here we have our distinguishing biochemical tests. Historically, they are catalase positive, oxidase positive, and DNase negative. And I'd like to draw your attention to oxidase and DNase because historically they are never found to not be oxidase positive. So always oxidase positive and DNase negative. And then once you identify the genus, individual species can be teased out through the carbohydrate fermentation profiles, which vary among the species. So was to characterize and confirm FO7DVA. And we did so by two different techniques, or we're in the process of still doing so. First technique was biochemical testing. And that's a very historical method, classic technique. It's culture dependent. You need to grow the bacteria and then subject it to certain tests or observe its growth characteristics. And then you can create a reaction profile and then compare this profile to published literatures and then devise, you can compare and then figure out which strain you have based on the reaction profile. Genomic or molecular profiling is a more modern technique in which we can use culture-independent methods potentially. You can also do culture-dependent, but culture-independent where you can grow it up and then sequence the RNA there and then use that sequence, plug it into a database, and then compare your sequence to known sequences and try to identify phylogenetic relationships that way. So for the general protocol of this study, we started with primary literature search, but I'd like to draw your attention to the fact that that was prevalent throughout the entire study, since this was a very historically based study on the history of Neisseria identifications. We then just did standard FO7DVA cultivation, biochemical differentiation, and we're currently in the process of the genetic analysis portion. So for culture media, we did general culture and thioglycolate broth. We also did Columbia blood agar, 5% defibrinated horse blood. And we did a chocolate tube blood agar, pre-made order from the company. We tested oxygen metabolism in a thioglycolate broth with a methylene blue oxygen indicator. We did motility tests in motility test media, which is a semi-solid media, and you look for growth deviating from the inoculation stab. And then we also did a pathogenic assessment, just to make sure we didn't have two of those pathogenic strains. And that's achieved through growth on modified Thayer-Martin 2 medium. And if it grows, it's generally one of those two pathogenic strains, but that media will select out all of those non-pathogenic strains. For colony and cellular morphology, we just did a standard microscopic examination and gram stain. Gram-positive cells appear to be purple based on this, and gram-negative cells appear a reddish color. Then we looked at the observable growth characteristics such as colony patterns and that type of thing, and pigmentation, and we looked on Columbia blood, chocolate blood, and also broth characteristics as well. For the biochemical test, we did treatment. Well, I'd just like to draw your attention to the italicized. I just wanted to remind you of what the Neisseria is supposed to be. Okay, so the catalase test was achieved through a 24-hour growth and then the addition of 3% hydrogen peroxide. Effervescence would indicate a positive test. The oxidase test is achieved through another 24-hour growth and then addition of the oxidase reagent here, and then a color change from like a pink to a blue-black or purple-black maroon color would be a positive reaction for Neisseria as well. And then DNase test is achieved through growth on DNase test agar, followed by the addition, or 24-hour growth, followed by the addition of 1N-HCl. And then you look at the growth, and if the areas of agar, they become cloudy, and if they're cleared around where the growth is, it's a positive reaction. So they are negative for that as well. And then carbohydrate, which is a phenol-red broth, which is a pH indicator with 5% fermentable carbohydrate. And if it can ferment it, the pH would change and it would turn yellow. So briefly go through the molecular methods. I'm kind of going to fly through them because they're pretty detailed, but we first started with the growth curve analysis that we've done so far. This is in order to get mid-logarithmic growth period, because that's when the RNA is the most optimal to harvest due to metabolic functions and all of that. cDNA generation comes, then we need to extract the RNA and purify it. And that we did through the kit, the QIAGEN RNA easy mini kit that I have there. And so this is the part we're at right now, and I haven't done anything below this, but we're planning on doing cDNA generation, and we're going to use random hexamers to do that because we don't want to exclude any sort of genetic material. So we're going to do a total cDNA conversion, and then from there we're going to select out the 16S rRNA using these specific primers here. These are known 16S rRNA universal primers that have been tested extensively and found to be very, very efficient at identifying the 16S rRNA genes. And then we're going to amplify that, and then following that, we're going to do some sequencing with a LiveCore 4300 DNA analyzer. That's just a piece of equipment. And we're going to use a 6.5% polyacrylamide gel to achieve this. Then we're going to run that data through a Saga, the LiveCore Saga software, which will then convert it to FASTA format. And then we'll be able to use the database that I was talking about earlier. We're going to use BLAST, which is Basic Local Alignment Search Tool Program. And we can just copy and paste our sequences in there, and then hopefully we can identify some sequence homology. The parameters that we're going to be using, greater than or equal to 97% sequence homology, will be identified as the identity of that isolate. Less than 97% sequence homology will be identified as a potentially novel phylotype and will require more robust testing to figure out the phylogenetic profile. So now I'm going to talk about the results. Starting with the biochemical results. These were all the uninoculated controls, so just take note of what they look like. Here's colony growth on chocolate 2 agar and Columbia blood agar. You can see that it's non-pigmented growth. These individual dots signify the colonies. They're circular, small, and then if you disrupt them, they're very friable. So that was another characteristic that I noticed. This was the result of the gram stain, so we were able to confirm that it was in fact diplococci gram-negative. You can see here where they meet in the middle. And that's often referred to with this specific genus as kidney bean shaped, which I kind of recognized here, so that was a good indication. Here's the results for the motility test. You can see it's clearly non-motile because growth does not deviate from that inoculated stab at all. The uninoculated on the left here, same here. And then here, these results are a little fuzzy, they're not that great. But here would be the aerobic zone with the methylene blue oxygen indicator. And we can see that the majority of the turbid growth is actually right below that aerobic growth zone. So with that and with the previous results for anaerobic and aerobic growth, they did a little more rigorous testing. We were able to confirm their results as well. So here's the catalyst reaction, and you can see the bubbles here where the addition was, and that's a very strong reaction to catalase reagent, or I'm sorry, hydrogen peroxide. And then here we have the oxidase test, and I put quite a bit of oxidase on here. And just to let you know that I did this multiple times because this is a very important test. So here's the results of the DNase. This is the 24-hour growth without the addition of 1-NaHCl. Here is the after addition of 1-NaHCl. So this test is a little bit tricky to interpret as well. I actually presented this information at a conference, and I did get some feedback from some other higher-level microbiologists as to what they thought their reaction was. And I think the consensus was that it's a tentative positive. So you can see here is the cloudier side, and the picture isn't the greatest representation, but here's the cloudy spot. And then right around the growth, you see where the agar is a little bit more cleared. So we had a tentative positive DNase reaction. And here's the carbohydrate profile. We were positive for fructose, dextrose, and sucrose. And no CO2 production or gas production in any of those. So, and analyzing the results, these are all the results that I just spoke up to about here. And once we get to the catalase test, everything jives with the identification of a Neisseria. But once we get to oxidase, which is a very important test for a Neisseria, it's usually the first test they list by saying that it's positive. We actually had a strong negative reaction. So based on that, I can preliminarily conclude this is not in the Neisseria genus as previously suspected. Continuing on, just a little bit more testing to get a better idea of what we're dealing with here, we did a DNase test. And the reason we did DNase is because there's another genus, Moraxella, or it used to be called Brandamella, but it's called Moraxella cataralis, the species specifically, that has historically always been misidentified as a non-pathogenic Neisseria. And the distinguishing test between non-pathogenic Neisseria and Moraxella cataralis is this DNase positive reaction. So that as well allowed me to kind of provide a little bit more evidence that this is not a Neisseria. And then this is kind of nullified here because we would use that for species differentiation, and since we don't know a genus, it doesn't really provide any sort of information. So I wanted to show some papers, like historical papers, on where I got the DNase information. I'd like to draw your attention to the date, 1988. It's not the most current paper, but that kind of shows you how out of date and hard to do biochemical interpretations are. So you have to work with what there is out there, and this is some of the most current literature on biochemical differentiation. So as you can see here, we have all of the non-pathogenic strains from here down. They're all DNase negative, except for, it says B. catarrhalis because it used to be in the genus Brandamella, but that's Moraxella catarrhalis. And then this was just the modified Theramar medium. You can see the pathogenic Neisseria grow on that, but they use multiple other mediums there too, so difficult to distinguish. Here's just another representation, and I thought it was interesting that they actually teased out Brandamella catarrhalis beta-lactamase positive and negative because that enzyme has to do with pathogenicity. And so if you can't distinguish the difference between non-pathogenic Neisseria and a potentially pathogenic strain, you've got quite an issue. And again, that is DNase positive. So we conclude that it's tentatively not a Neisseria, but we don't know what it is, and we need to do that molecular testing to really figure it out. But conclusively, we can say that culture-dependent methods aren't reliable. They're very hard to interpret. As you can see in the DNase test and a couple of the other ones, you don't always get a very, out there reaction. It's not always exactly what you expect it to be. So there are multiple limitations, also the media that you have to buy, what you can culture, what you can't culture, if you don't have anaerobic conditions to grow your bacteria in. There's lots of different things. And then one of the most important things is that gene expression can vary. It can vary based on the media that you choose. You can choose really highly nutrient-rich media and grow your bacteria in optimal conditions. And that's great, but you're forcing a genetic profile, an expression profile that might not be the natural one of that bacteria. So you might be characterizing something or forcing evolution of a different strain. And in that relation to that, I want to talk a little bit about biofilm composition and why it is so important to not isolate these species and study them on their own, because it does change what you're looking at so much. So here's just basic organization of biofilm. Here's the two. We have food webs here where they actually have a lot of nutrient exchange going on and different processes like that. We have crosstalk between the host and the bacterial cells. Here we have a resistant group where this can be done two different ways. They can secrete enzymes that cause these bacteria to appear antimicrobial resistant, or the overall architecture can just, biofilms are usually coated in a rich polysaccharide film or protein-rich film that are historically antibiotic resistant as well. We also have genetic transfer, so horizontal gene transfer, which is another reason as to why these shouldn't be isolated, because if you think about pulling one of these out, it could have interacted with these bacteria here, and you have a whole different profile that you're dealing with. So yeah, it's basically good to study these, if you can, in their natural habitat. And so that's where the issues in our study arose, but there are ways to get around that. And a kind of new field that's coming out is metagenomics. And metagenomic methods entail taking environmental samples, so like a super gingival swab like we did, and then going straight to the molecular methods. Like you don't have to grow anything in between, you don't have to harvest RNA, and you can just sequence and then compare those sequences you do PCR and then compare those sequences to known strains. So that can give you a better, more accurate profile of what's actually there. And it also identifies, and I wanted to say one more thing. Just the difference that I had between what researchers in the same lab, in the same school, with the same exact strain had, just shows you how unreliable biochemical methods really are. So for future aim, we'd like to figure out what we have, and then go on from there. And so I'd like to say thank you to Dr. Fryer, my very attentive and patient advisor, and the funds that he gets from the School of Sciences to help support our research. Dan and Vicki, who did the original isolation, and then Slurpee, R&D. who we got the original cultures from. Dr. Farr's cat. Any questions?
What are the chances you find whatever bacteria you have on another one of Dr. Fryer's cats that in the world biochemical?
Well, I'm glad you asked actually, because while I was doing this, I actually just found a paper in print. So in 2011, one group came out and they were doing a total metagenomic analysis of the oral biofilm. But they recently just came out saying that they're pretty standard across the board on what they're finding because they did multiple cats. But they also are finding 2 completely novel phylotypes that are not anywhere. So, I mean, this could be one of those. Who knows? But since it's a healthy cat, I would like to assume that it has a standard profile, but there's really no definition for that term.
Other questions for Julia?
Where do you see this going? Say, let's look years down the road when the feline oral microbiome has been characterized.
Yeah, when that hot topic issue gets solved. So I see this being used in veterinary medicine. I guess I didn't know this, but a majority of the time in veterinary medicine is actually spent on oral health care. Cats have very prevalent periodontal diseases, dental caries, and things like that. So by knowing what's in there, we can potentially develop control methods such as nutrient inhibition, enzymatic controls, toothpastes, fish bones. I mean, you know, something that the cat can chew on, any type of physiological control to these, try to, you know, inhibit their formation. And if we can get at those initial colonizers, that's the even more important part, is that we can stop the growth of the biofilm completely.
Would that be a good thing?
Well, it could be a good thing if the biofilm is causing the illness, like a periodontal disease, but it could also be a very bad thing because a lot of times, like mucosal biofilms or intestinal biofilms and oral biofilms even are protective, and they provide, like I was talking earlier with the immunological protective actions. For lack of a better way to explain it, they take up room so pathogenic bacteria can't get in. So, yeah, it could be bad to control them as well. But as you know, one of the late colonizers, well, you might not know this, but as one of the late colonizers of biofilm is P. gingivalis, which causes gingivitis. So maybe hitting it in the middle of the formation, I don't know. But.
Are there particular conditions then that you might think that management of an oral microbiome would be more important than in others, or in a normal individual animal?
You mean like if conditions of the animal themselves?
Yeah.
I would say yes if they have any sort of condition that causes a suppressed immune system, because a lot of times the commensal bacteria are opportunistic. So they're not generally pathogenic, but if you have any sort of compromised immune system, they can opportunistically cause infection. So I would think, you know, in a cat, say with FIV or something like that, you would want to control the biofilms more so than you would in a healthy individual.
Yeah, no, that's exactly right. That's what we check on for our other cat who has FIV. The way we evaluate his status is looking at his gingival inflammation.
Yeah, and I actually came across a lot of things that said it's an indicator of total health, is their oral health. So.
I should know this, but do cats get cardiovascular disease? I mean, for instance, mice won't unless you mutate the APOE gene, but will cats develop it?
To go off on a total tangent, can you clarify that question? Because that's a really broad...
I'm thinking of say, atherosclerosis and links between gingival diseases and atherosclerosis.
That I don't know. There's actually learned by experience from the other cat we took in at the same time we had Slurpee. Congenital cardiovascular disease is actual a problem, and I had never realized that. This cat had a severely enlarged heart, and the ventricular volume was, I was shocked this cat was alive. It was so tiny when I saw it on the scan. And the vet said it was the worst case he'd ever seen. And this cat just kept chugging along. We had it for 5 years after that diagnosis. I still don't know how it survived 5 years. So in some cases I would guess yes, but I don't think atherosclerotically,
I would guess.
but I don't know. I'm not that good a veterinarian.
Yeah, because there's human links between dental health and
Yeah.
Yeah.
Which is why if you've had certain types of infection, you're required to take antibiotics before you go in to get your dental work done. They don't want to risk you developing, what is it, pericarditis? Is that? Yeah. Yeah. Right, because when you clean teeth, it doesn't matter whether it's cats or dogs or us, you can cause small wounds and bacteria can get into that. So I know with cats, they actually put them, they knock them out.
Whenever anyone says bio, I can't knock them.
I know, now every time you run your tongue over your teeth, you're going to go, oh, I know too much about this.
Motivation to brush your teeth.
Yep.
All right, thank you.